Hello friends, through this video we'll discuss the scanning electron microscopes, their definitions, principles and procedures, application and their instrumentation. So the type of electron microscope that scan the surface of the specimen by passing an electron beam is called as scanning electron microscope, also known as the SEM, and it was designed by the Max and Knoll in 1935. So SEM is very useful to study the surface architecture of cell membrane organelles and others. So principle: the SEM uses an electron beam as illumination and electromagnetic coil to direct the path of electron beam, and when electron beam is focused on the specimen, it produces three types of beam, that is secondary electron, backscattered electron and the characteristics X-ray. Instrumentation of the SEM: it consists of the electron gun. So it is a V-shaped filament vanillate cylinder and cathode and anode plate with a hole in the center. So the figure of the electron gun is as follows. So in this figure, it is the cathode shield, also known as vanillate cylinder, the tungsten filament, and it is a wire which is attached to the high tension volt, and it is a beam of light passing through this type of hole. So second one is the electromagnetic coil, also known as the condenser lenses, consisting of two in number. So deflection coil just below placed in the second condenser lens. Next is the specimen stage, placed just below the deflection coil, and it is in slanting position in the electron path. Next is the electron detector, and each detector is composed of the collector, scintillator, PMT, that is photomultiplier tube, and it is of three types, that is secondary electron detector, backscattering electron detector, and the characteristics X-ray detector. Next is the photographic plate, also known as computer. The image may be captured on the computer or hard disk. So the SEM is heated to a great extent in order to keep the apparatus at a low temperature. Cooling water is circulated through the cooling system around a SEM. So it is a system of the SEM. So in this system, firstly the electron gun is placed. Then two condenser lenses are placed, and then just below it objective lens are placed. Then this is a deflection coil and objective stage are placed here, and then the passing of the beam of a light, an electron beam is passed through this hole, and then it is both the deflection light, deflection coil is just connected to this secondary electron detector, and then X-ray detector is placed in this manner. So system of the scattering electron microscope. So the dry specimen, which may be the wood, bone, feathers, insect or wing. And for the wet specimen is fixed by immersing it in a solution of the fixative, which stabilizes the molecular organization as such in them by forming covalent bond. Fixative like the osmium tetroxide, glutaldehyde, potassium permanganate, formalin or the formaldehyde. So it is used for the wet specimen. And then the application of the SEM, that is, it is used in the virology, investigation of the virus structure, 3D tissue imaging. For forensic uses, it is used in surface of small archaeological specimen or the fossils, gives topographical imaging, surface feathers of an object. So this is about the SEM, scanning electron microscope, known as SEM. So in next part we will see the TEM, that is transmission electron microscope. Thank you.